other languages experience, then you may want to look at the difference between this language with the language you are familiar with. So this tutorial is what I was talking about, this bottom up. So you learn from the very basic, from the desktop basics, and move to the features of all the language. How to define a matrix, something like that. Yeah, so I think I will let you to work on this for 10 minutes while everyone is installing. And if you don't have this, you can Google, because this is a website, and found this and start to read some basics. You see my Google search, the first hit will be exactly the same page you will see from the inside documentation of MATLAB. And here you have a web-based tutorial, and just go through them a little bit, and then we move on to the top-down method. Still waiting for the installation. Still, it's done. Good. And remember this ZenWorks interface, because later we may want to install R. So we run two languages side by side, and they communicate to each other on the same machine. You can do it right now. But not today. Today we don't need R. So in MATLAB, on your right top, this button, Learn MATLAB. So that's your resource at home, and you can click and access to a large amount of this tutorial documentation and the video. Okay, so on this interface, in the middle, the large white area is your command line window. Make sure you know where you are. So that's a working folder. Okay, in R, you have a working folder, and here is the same working folder. So if you have any function writing in the script file, function file, you should be able to find from this current folder. If you have some other folders, see other folders. So if you have other folders, any files function inside that folder will not visible for the program. You have to add this into the parse, search the parse. So anything you hide under that folder will still be readable from the current working folder. So you just have this concept. Otherwise, you will see, okay, I save my file, my function in somewhere, but how can I call it? Because the program cannot find it. All you have to do is add the parse. So the subfolder will be accessible for the program to read the function inside there. So that's the basic concept. Okay, say you will clean this. So if you type 1 + 1 and it give you answer 2, and you can see A equals 1, then semicolon, then you replace this feedback. So if you don't put the semicolon, it will print out the output on the screen. But if you put the semicolon, you splice the display of the results on the comment window, and the B2, A plus B, the answer is 3, something like that, okay? So if you decide to type in those matrix, then something like this, you can type those in. The operation is based on the matrix, so you can A plus 1, every element inside just plus 1. Those are the very basic operations for the matrix number assignment. Extract column rows will be similar to other languages, right? For instance, I can see the second column of the A. Okay, so A is this, the second column is 2, 3. Second row is, okay, so you can work it out. For those you familiar with this, so that's too basic for you, you can take a break for a couple of seconds. What you can do is to, for example, found single cell paper and see, looking for the data set you want to get in here and see what we can do. I know 2 of you study bone. Any good paper around the bone genesis? We can look at it together and see how can we get in there. So we start to do analysis directly, okay? Database is powerful because it comes with a lot of toolboxes. So for example, if I search regress, the function where it is, it's located inside this version of MATLAB, inside this toolbox, statistic toolbox, there's a function called regress. You don't see anything here, but because you have a parse, which already include this parse into the searching directories. So you should be able to access this function directly from anywhere in the environment. So if you want to know more about any functions, you can see doc or that function name. And it will tell you how to use this function. Okay, what's the input, what output. So that's a list of the toolboxes. We know it's engineering computer language, so you see a lot of this. Engineering related tools, control systems. And we do have a bioinformatics tool. And also, if you brought it down, and the statistics and machine learning will be the most important toolbox we are going to use. This is a really very good reference book, actually, if you want to browse through and their examples, their explanation. It's encyclopedia, okay? So it's technical documentation. Any questions so far?
Yeah, I actually had a question, Dr. Kai. Yeah, I was wondering, were we supposed to have installed the stats toolbox from somewhere? Because I don't have the regress command on my laptop.
Yeah, Alex, so your question is, on your machine, you don't have those toolboxes installed?
Yeah, those two functions that you were just using, regress and PCA, I do not have those.
Yeah, that's because you don't have a statistics toolbox installed. Let me show you how can you get it. Okay, so from here, you see my screen right now. There's
Yes.
a button here, add on.
Okay.
If you click this, see get add-ons. So those toolbox has been described as add-ons. If I search You see on my screen, can you see this?
Yes.
This toolbox has already been installed. But on your machine, because your own license may not include this, I'm not so sure.
Okay.
Maybe not pre-installed this, but you can check
Okay.
and just install it.
Okay. Thank you.
This is the only toolbox we will need to do our analysis. There are many different toolboxes we don't really need. We don't need a bioinformatics toolbox. Okay, I don't know if you are on this page now. If you search single cell RNA-seq, you should be able to get the toolbox we are going to use. Because if you don't have this installed, all the function I wrote will not be accessible to you because you haven't installed them. But if you decide to install from here, you can give it a try. I'm not so sure because I'm going to download the developer version, and but this should work as well. So this is number one, the one way to install Toolbox. So the first is using the add-on. To install Toolbox as add-ons, and another using the command line window. Okay, download, direct download. This is good because you can uninstall anytime if you don't like it. The reason is this will install into your local folder. You don't have a control where it can be installed. But the direct download, you know exactly. I want to download in my working folder right now. I can see it, change it. That's only difference. So I'm going to give it a try and install using this add-on, and then I will uninstall it later. I'm not so sure whether it will work. Oh, you need a. So I gave up. I do have my account. But you know there's option like this, okay? So now we have nothing, there's no function to help you to analyze single cell RNA-seq data. So we need a toolbox. We need functions, a lot of functions per writing. And what we can do is to here, let's Google search. Gene expression analysis toolbox. So that's a keyword. So that stands for single cell gene expression analysis toolbox. On my screen, the third this link will be good because you will have the code for you to install. Okay, on this page, you just click quick installation, then that's the file, not the file, that's, you can copy those and paste into the command line, and it should go there to download and install for you. So, remember, my working folder is here in my documents, MATLAB. So my toolbox will be installed inside here, and the parts will be added. Yeah, let's see no right now because the last step, add the parts probably has some problem. Well, unable to save the parts. Yeah, so that's fine. That's fine. So that's why I save this option. So because we don't have the control for the as admin, so you cannot save parts to the systemic folder, but this one will allow you to save your user parts. So you copy this and put it. Just one more step. We are done. Okay. So you can see this installation. This procedure is transparent. It just go to that website, download the package, unzip and put in the local folder. Then add these parts in here. And then next time I don't want to re-add manually, so I save parts. It doesn't work, so I save to the local here. So next time I don't have to repeat those. Already everything is in here. Okay. With this, we can start to play with data. Okay. I haven't told you how the data get in there or the program, but we from top down, we start to ask our questions. So I want you to bring the question to me and which paper I should look, whether the paper has the data, has they provide a session number to the data. So all I need to know is a session number. The reason I say we can do data analysis straight away is because inside this toolbox there are hundreds of the functions already support this. So I can comfortably say, okay, we can do analysis right now, even though you haven't coded anything yet. We can see how far we can go. Okay, we'll see. I remember you have a paper about the B cell germinal, yes, germinal affinity. What's the title of that paper? Restricted. Yeah. So I'm going to use an example paper and see how we can, because Eric has a project related to this, and someone else published the paper. And we think, okay, the data is interesting, and we should take a look ourselves and download the data and play with it. So how we can do it. Eric gave me the title of the paper, so I look at it. So this published in Immunity, so this high profile impact factor journal. They study, I think that's a mouse study, and they have some kind of a cell sorting and to isolate particular type of the B cell. And there are animal studies. Okay, of course, it's better you understand this and read the paper, but for now we just jump to look at the data, okay? So I have to browse this down and see whether they have single cell data made available. So it looks like this is a single cell data because you see the UMAP and a cluster of different cell types. So they definitely have their own data set. Yeah, so this cell price, this publisher, they provide this methodology part with all the resource, including data set. We should be able to find the tags. We have reagents, we have plasmids, software. Yeah, here we go. This PDF have 41 pages. On page 21, I found the ID. So that's where they put single cell RNA-seq data. This paper generated. This is the NCBI database. Okay, we need to know this and visit this page. I copy the ID and the search, and this is the record in Geo database. Okay, you see the title, you see this. And that's the right paper, and there are two samples. to animals. You don't see any matrix file here. So we have to go inside the individual sample. So I click one of this. So this is so-called WNV. I don't know what it is, but it's a wild type and treated. There are some B cells in there. So I look at this record and browse it down and see, okay, there's 3 files. So this is a typical organization of data file generated from 10x genomics. You have 3 files. One is the matrix in MTX file format.
It says there's a series matrix file. Is that not the same thing?
Mm-hmm.
Let's see if that's that one.
Previous page. Okay. No, this is so this file has been there for many many years. From a microarray age already there. I think that's the indicator of the sample ID, something like those. They're not a matrix file. Mm-hmm. FTP side, yeah, some linkers and so they're not. Normally they should be in here. Depends on when the author upload their file, whether they want to separate it into independent samples or they want to put together, put in a joint file here. Sometimes you'll find a joint file right here. Yeah, but in this case, we have independent 2 samples. Each sample has 3 files. So this is a matrix, this is the gene, and this cell name, 3 files. Remember, we have a matrix, and then rows are the genes, and the columns are the cells. Cells is barcoded with the 10x genomic barcode, each cell in each GeoBead. So there you have ACGT base pairs in there. Okay, so that's 3 files are what we need. Normally, if you you can download them and save you in your local file and then read it. That's another way is we can do this automatically. So let's select this ID first. Oh, I can click here and copy this. So I need this sample name. And then we come back to MATLAB here, where we use single cell gene expression analysis tool. Okay, we are going to use this to do what I just described. We will download 3 files and load into here. But now I can do this automatically because I have this function which allow you to automatically go there and grab 3 files and read in there. So we will finish this step before break. Then we will stop there and take a break and come back. Okay, so there are different ways you can get data into here, and the geo accession number is one of the ways. So I just select this and click. And here I will provide the ID I just copied. Okay. I click OK, and then I will go to take a break. Come back. Okay.
Where is it installing this data to? Is it going to be local or is this?
Alex, can you repeat your question?
Yeah, I was just wondering where is it saving this data to? Is it going to be somewhere saved within MATLAB, or do we have to re-download this every time we start it up?
No, the temporary file was saved into the temporary folder of your system. So, yeah.
Okay.
Okay, so what you see on the screen, this each point is a cell, and the information is from this matrix file. Okay. So this matrix is 30,000 something how many genes and 4,800 cells in this matrix. The position of the cells are randomly arranged in this three-dimensional space, because I just want to give you impression there are a lot of cells in there. Okay. The relative position of them are randomly assigned. So this cell may have nothing to do with the cell next to it, because we haven't done anything to compute their relationship yet. One question for you is, on my screen, I can see, okay, there's one cell here, another cell here. Do you see exactly the same location? So though this is a random number, because we use the same random number generation algorithm, we're supposed to have generated exactly the same number. Okay, so random number is not a real random, it's a pseudo random number. Otherwise, we shouldn't see the same location of the cells right here. Okay, now is the power of MATLAB, this user interface. You don't be able to easily to do this using other languages. I know in R you just cannot do it. Here, if you select this rotation 3D, you can highlight, click this, and you can start to rotate your data. Okay. You can brush and select. So if I don't like those cells, I can select and do something for them. I can data tips, and I can see, okay, what's the value of this? I customize this display function, so I only display the category of the cell. They all in the same group. You can save, you can take a picture. Okay, a lot of things you can do. Now, if you highlight those buttons, which I put on as a shortcut, you will see the basic function of each button. The first one, for example, is to select the gene and show its expression. So let's see how it works. Here we have 31,000 genes. Do you expect so many genes expressed in individual cells? Human has 20,000 protein coding genes, right? So this list, this data is from a mouse. I think this list is a annotation. All the genes annotated, provided as a reference. So even though maybe some gene have all zero expression, is still kept in there. So we have to do some filtering and see. No expressed gene we will get rid of from the list because we don't have to analyze them. What we can try is first want to look at the expression of these genes look at individual gene, for example. And I can sort gene by alphabetic, or I can sort by average expression level. Let me try to see sort by expression level, because I want to see how low the expression can be for the gene to be included in this. So on the top is a highly expressed gene. So you can see mitochondrial genes, okay? You can see a lot of housekeeping genes. Makes sense, right? I want to browse it down to the bottom to see, okay, this gene. I click OK. No expression. If there's expression, then one of the cell will be highlighted as a red. Okay. In this case, everyone is in light gray, 0. So we definitely need to delete that gene now to let's see another example, see highly expressed gene. I will select something in the middle of here, see. Okay. So this gene expressed in 25% of cells. And that's expression level. The level is measured by, remember what? The number of UMIs. Okay, we mentioned the UMIs. It's not the amplified number of copies, it's unique copies. In this case, you can also rotate the files, your picture, and in order to give you a better angle to see this stem plots, okay? By the way, you can click this link, and this will link to the gene card, gene ID, and to look for the name of the gene and other information. Okay. Play with it and see if you can have any questions.
I'm getting an error when I try to look at it. It says I'm missing a function grp2 idx.
Missing a function.
Or yeah, it says I have an undefined function for.
Yeah, so you need a statistics toolbox, remember?
Right.
You miss one.
I-
So that function is inside there.
Okay, because I'm pretty sure I installed it because it had to restart my MATLAB, but I can check and make sure that works as well.
Yeah, just do a search and see where this function is located on your machine. Make sure you add this parse so you can find it and use it. So statistics toolbox is only toolbox we rely on. There's only one dependent. Okay, if you want to see multiple gene expression altogether, you can have a, say, I want to select multiple genes. And then look at its expression individually. Okay, so what do you think what next step we should do if you have the data like this? Yep, you can do that directly, but I want to get rid of those non-expressed genes first. So this 30,000 gene is the number is really too large. Normally, less than 10,000 genes should be reasonable by 10x genomics technology. So we're doing some filtering right now, and the filtering will be this, okay? The third icon here, right here. There are different options, but the first one already been set up. There are different combinations of some parameters we want to use. We may spend this sometime later, and then you have time and go through the other stuffs. You can use a basic filter to see I want to use more relaxed or more stringent cutoff. More stringent cutoff means I require, for example, a gene to be expressed in more than 5% of cells. If it's lower than that cutoff, I will get rid of them. Or if you use only 1%, that's more relaxed, you keep more genes. I use it. So that's a different cutoffs for the QC. Library size means if you look at a cell and you summed all UMIs in total, how many total UMI for that cell? If you have a large cell, then you may have a larger UMI, total UMI summed. If it's a cell poorly sequenced, and maybe UMI total number is too low, you know it's not well captured, you want to get rid of them. So that's the setup for the threshold. The mitochondrial rates ratio, so mitochondrial against the total rates. You don't want to have a lot of mitochondrial rates in there. That's an indication of the cell damage, cell membrane break, and you have a lot of multiple copies of mitochondrial genome transcriptome released. That's low, so that the high number of this is indication of the low quality of the cell preparation. There are others. Okay, so I just use a very relaxed cutoffs. Okay. So this option is for people to experiment. A lot of them say, okay, even though there are some genes only expressed in one cell, I still want to keep it. I want to see where this cell is. So I don't want to apply any cutoffs on this gene. So I provide this so you can keep that white list of gene. No matter how bad the gene is, it's still there at the end. Normally we just say no. Okay, this procedure will automatically filter cell first, then gene, then iteration, cell again, then filter gene until there's no cell and the gene be discarded. Okay, so using this relaxed QC cutoffs, we removed 378 cells and more than 20,000 genes. Okay, so now it's a better number, so we know it's clean data. So now I look at the lowest expression gene. You still have 1% of the cell have this. Okay. So as you said, we should start to group cells into same position. So that's the algorithm right here, and it's called embedding. And just select TSNE. I suggest one of us use this 2000 highly variable genes, another use all genes and see whether we get the same kind of embedding. So I will go ahead and use 2000 genes. Yeah, so that's my 2000 gene. Similar, same. Okay. By the way, t-SNE is a stochastic process, so this time we don't expect exactly the same. Maybe still, I don't know if you have this. Same, okay. Cells use gene as a feature. Yeah, to look at the similarity between cells. Oh, you get this error message.
I had it open.
Oh, yeah. I think it's in, if you minimize this window, maybe you found somewhere else. Oh, no. Okay, let me show you how to put it back. So if you lost it, single cell gene expression analysis tool, bracket, this one here, and then you put SCE in. SCE is your data. You should be able to bring it back. Okay.
Thank you.
You also have SCE1, but that's another data set maybe. Yeah, but yeah.
Thank you.
No problem. Okay.
Messing with it in my computer. I'm like,
That's okay. You just
"Oh,
do that
I have to
again.
restart."
Yeah, it's okay. If you crash the computer, just restart.
Yeah, you'll laugh. You color my groups, like the different colors of the cells. Thanks.
So once you have this, what's your next question? Once you have this. Why do you want to grouping them? No.
Cell types.
Cell types. Okay. Yeah. Sometimes my first question is, what's this, like this group? Why it's isolated, becomes small island by itself, right? You have this question here. Let's see. I want to see. Yeah. So one of the way you can check, see this group. So this group, why itself is so far from the main island. I will click this marker genes for the brushed cell. So I want to find the genes highly expressed in those cells only, but not expressed in others. So that first will give me some clue. Okay, what the cell type is, right? So I will do that just for fun. Okay, I want to have a top 10 genes. No. So this top 10 genes, now it's your turn to use your biological knowledge. Okay. You see CSF3R, I don't know. I have to check. Okay.
So does it do just the 10 most highly expressed genes?
Highly expressed gene exclusively in that your selected cell. Yeah. Oh, the button I used was, once you select cell, then you use this. Okay, this 10 marker genes, the list is here. And that's exactly the knowledge, the database search you want to use to see, okay, I will use this marker genes to search my database and to find, okay, this is most likely to be, I still don't know because I don't have knowledge of this. Is that NK cell maybe? Neutrophil, yeah. Here we go. We have a cell biologist around us. So instead of a search for the markers, I also provide a function here. I use hemoglobin genes and I look at which cell has a high hemoglobin genes expression. I want to find the blood cells because those cells will be the contamination. So this procedure is opposite. Instead of a search for the marker genes, I use no marker genes to search which cell has a high expression of hemoglobin genes. So if you click that, you will see whether we have blood cell contamination in the cells population. We do have some, maybe. And yeah. So this is a relatively clean data set. Okay, some of the primary tissues, you see a lot of blood cells. They're not very interesting actually, right? Because we know their function so well. So they're not involved in the. So this chart is a combinations of quite a lot of different blood cell marker genes, to tell you. Okay, so this manual annotation is slow, but sometimes it's very useful, especially when you exploring some unknown tissues. You really want to know this group, that group. So you have to use your biological knowledge to judge whether this is a peculiar part of cell type. And you can select not only 10 marker genes, but the top 50. So once you have a top 50, you should have rich more than enough information about the cells. So instead of, yes, the questions. Yeah, so that's the way we use expression profile to find the similar patterns which we know using this profile to assign the name of the cell type. So when we add some, for example, add some blast and the expression of the
they change. So can we think that this drug they change the cell type?
Yeah, if you have a drug, let's see, I want to make cancer cell become a normal fibroblast. Yeah, certainly, that will be cool. Or you even push cancer cell towards the fibroblast or epithelium cell, normal epithelium cell a little bit. Yeah, that's what we are trying to achieve. Yes. And in stem cell development, you certainly can induce stemness of cells to make them become stem cell or develop back to mature cell types. Yeah, we do have those transcription factors cocktails to help you to do this. Yeah. Okay, so it's a good way to do this manually. I certainly encourage you to do this. You can click this and see assign into a new cluster, into a new group. So I make 2 groups and make 3 groups. You see this? This is separate into groups. Yeah. Then you can annotate individual groups by give a new name, something like that. But normally we want to do automatic clustering. So here there's tree-like stuff. If you click this, we are going to use k-means to automatically cluster cells into different groups. Okay. And normally we will overshoot. See, I want to do 50. I break down cells into a lot of small groups and then annotate their cell types. If they are same cell type, I will later group them together as a merge them as a larger group. The larger number here, so we figured out is good because you want to have an independent cluster for this small groups. For example, you want to have a small group for this, small group that, and annotate them independently. And if they are same, later we merge them. Otherwise, we can separate it. If you don't overkill by make them into small cluster, you say, okay, this is one cluster. Then you have no chance to give independent annotation if they are indeed are different cell types. So that's why I suggest we put a big numbers here. So you separate the cells into a lot of small clusters. Okay, and then the next one is to click that button I'm just pointing to. That's automatic annotation of cell type. Now you just wait. Okay, it will annotate all the small clusters. The sample is from a mouse, so make sure you select the correct. yes or no. So right now, we may want to merge, okay, merge the cell. You don't want to have a 45 different, 56 different subgroup of B cell. Of course, you want, yeah, maybe later we further subclass them. Okay, we are done with the annotation. Yep. Rename it. You can see I want to rename. Yeah, I have a database built in to search the database. Yeah, for example, I know B cell, their gene A, B, C, D, E should be highly expressed. And then I have a algorithm to assign relative value for each cell population. And then yeah, we will discuss this in the middle of the semester. Okay, how exactly it should be done? Expression.
Because there's so many.
Yeah, yeah. Remember the neutrophil file we talk about. I think the correct annotation there. So what's the next step? So this group in the middle, this the red blood cell. So I will remove this. Okay, delete them. There are two ways you can delete those cells. One is by select them. So if, for example, I select all these red blood cells, then delete select cell. Another way is to select all other cell types and make a new data set. So it's this button. If I say, okay, I want to select cells by cell type, and I select all the cells instead of red blood cell, and then this will produce a new data set without the red blood cell. Okay, either way. So at this stage, I'm going to save my data, and for the next time I use. To save your data, click that button, and then see, I want to save to MATLAB. MATLAB is MATLAB specific data file format. So everything, all the information will be compressed and saved in there. So I can see, I want to save this to my current folder, give a name, so my data set number 1. Okay. Now I'm going to shut down this and see how I can load back the file I just saved. I see, okay, I close this. No. Close this, not save. So I'm going to clean up everything. If I don't clean up everything, you see here are my previous variables. Remember, I tapped A, B, C here, and that's the data we read from the download from the website. And now I just clean them all. My data set has been saved into my working folder currently. That's that file, so that's in there. So how can we load it back? Just tap load my data file. So what happened is the variable inside that file has been loaded into the memory here. So that's the object. It's a container, and all the information we just annotated, and it's already constructed in there. So that's the file, and now that's the variable, and there's a structure. And if you double click, you should be able to see under this variable there are different sub properties. It's actually the variables as well. So the X is the expression matrix, big X. This is the gene list. S is the position of the cell in the 3 dimension, and the C is category. So you have a lot of information about those. So this object is here.
So I feel that my computer like froze for a while.
Corrupted the file.
I don't know.
Yeah. It's not in your folder here. You can double click that file and it's saying
It says error using open line 143, not indexing
Yeah, so you get it. Good. Oh, you saved it. It's not a data file. Mhm. You save the figure. It's not a data. Mhm. What we can do is to, if we have a shared folder, I can drag that my file here, and then it should be. Let me think. Yeah, if you have a USB, something like that, I can transfer the data next time to you. So
Okay.
today we are almost done.
Yeah.
Okay.
Swap the-
Yeah. You saved the wrong file. I mean. Okay, so when you have this in the computer memory here, you can SCE itself is a variable. You can see it. To load it, you use single cell gene expression analysis tool and SCE in here. So you can load it back. Using this. If you don't put anything, remember, if you don't put anything, you will be giving this. You have to redo it. But now because you have your variable already supplied to this, it will give you this directly. Okay. Everybody get it? Yeah, good. To label the categories, you select that button and select the cell, the labels you want to put in. So in my case, it's cell type. Okay, the last function today we will be talking about is this button, and to make a video, basically, then you share with your collaborators with a MP file, MP4 file. Okay. Yeah, so the generated file is inside this folder, and this is double click. Okay. All right, that's all. And questions.
How to delete the cluster by cell type again? I missed it.
Okay.
Figured out on my own.
Okay. To make clusters, click this. use one of the method, and this with the rabbit is much faster. And I recompute. Okay, I tend to overclassify them, cluster them, because I want to have a subtle details of the small clusters. If we don't, we do something like 20, then that's clusters. And then automatic annotation is this one. If manually, you will have to select from the best guess, but this will just give you the top, the most likely cell type. Okay, that's all.
Oh, I want to know how to, I didn't delete the erythroid cells. I want to know once you get there, how you delete the.
Oh, deleted some cells. Okay.
Without having to just go in manually select them.
Okay, for example, if I want to delete this group here, I just brush it and say delete. Delete select cell. Okay, delete this. You can.
But there's not a way to go in and say, tell it delete cluster 5 without brush selecting it.
You should be able to. Let me see. So I select some of this in the group, and I will see. Okay, select. What I would do is to.
Yeah, select cells by class. That's what I was looking for.
Yeah,
Exactly what
I
I was
see.
looking for.
I select all the cell types I want to keep.
There we go.
Yeah, I select all the cell types I want to keep and remove NK cells, something like that.
Perfect. That's exactly what I needed. Thank you.
Yeah. It's not a delete. It's a select subtypes and make a new data set and save. Okay. You can ask me question anytime through the email during the day, during the week, and otherwise I'll see you next Tuesday in that room. Okay.
I think it's because there's. Is like for some classes, and they use I think that there's just too much stuff on this computer, so that's why they're so slow. So